Doing paper 1-3 yet simply cannot understand one question? You clicked on the right video. Welcome to my channel where students of biology are simply interested in learning more. This right here is a 9700 May-June 2020 paper 13. If you are new to this channel, you know what to do. Hit that subscribe button. Without wasting more time, let's then jump straight into the video. Question 1. A student was given a photomicrograph of a cell and told the magnification of the image. The student was asked to calculate the actual size of the cell. Which row in the table explains how to do this? Now the question asks us, how do you calculate the actual size of the cell? And if you recall the formula for magnification, magnification is equal to image size upon actual size. Hence the actual size is equal to image size upon magnification if you rearrange the formula. Now if you go through these rows and columns, one column says rearrange the formula to. Now since they are asking for the actual size, it should be I over m. Therefore the only answer that represents I over m is option C. Hence the answer should be C. Question 2. The electron micrograph shows part of a eukaryotic cell. Which cell structure is the site of protein synthesis? Now protein synthesis occurs in ribosomes and in the RER, which stands for rough endoplasmic reticulum. Ribosomes are represented as small black granules in this electron micrograph. Therefore, the answer is B. Question 3. Some stains can be used to identify cell structures in living cells. A dilute solution of one stain causes the whole cell to appear blue. The blue color rapidly disappears in most cell structures. Those cell structures that release energy stay blue. Which type of cell structure is likely to stay blue? Now I have highlighted the key part of this question. Those cell structures that release energy stay blue. And they are asking which cell structure is likely to stay blue. Therefore, they are asking which cell structure is releasing energy. If you go through these options, mitochondria is the only organelle that releases energy or produces ATP. Hence the answer should be D. Question 4. A molecule of carbon dioxide is in the center of a mitochondria. Assuming that there are no other cell structures in its path, how many phospholipid layers will the carbon dioxide molecule have to pass through in order to leave the cell? Now this question requires a bit of thinking. We have to consider not only the mitochondria but also the cell. Now the mitochondria has a mitochondrial envelope, which is a double membrane. Since it is double membrane, it has two membranes, and if you consider the membrane of a cell, it will have altogether three membranes, and one membrane is made up of a phospholipid bilayer, which is two layers of phospholipids. Hence the overall answer is 3 times 2, which is 6 layers. Hence the answer is C. Question 5. Which row identifies the type of ribosome found in each of the different structures? Now, the type of ribosomes found in chloroplast and in mitochondria as well as in prokaryotic cell is 70S, and in eukaryotic cell it is larger, which is 80S. Therefore, if you go through the options, the answer is B. Chloroplast 70S, cytoplasm of eukaryotic cell 80S, prokaryotic cell 70S, mitochondria 70S. Question 6. The color of a positive Benedict's test is due to the formation of copper oxide. The mass of copper oxide is proportional to the mass of reducing sugar present. Samples of fruit juice were tested for the presence of reducing sugars and non-reducing sugars using the Benedict's test. The table shows the mass of copper oxide after boiling with Benedict's solution and after acid hydrolysis and boiling with Benedict's solution. Which sample contained the most non-reducing sugar? Now the test for reducing sugars is generally done by carrying out the normal Benedict's test. Whereas for non-reducing sugar, you first have to hydrolyze it using acid and then neutralize and then carry out the Benedict's test. Now they are asking which solution contained the most non-reducing sugar. They have given the table and they have given after boiling with Benedict's solution and after carrying out the non-reducing sugar test. In this question we have to consider the difference between the two values to get how much milligram was of non-reducing sugar. The largest difference will give you the most non-reducing sugar. Hence the answer is B, since B has the largest difference among the two values. Question 7. Which polysaccharides contain 1,4 and 1,6 glycosidic bonds? Now it must contain both 1,4 and 1,6 glycosidic bonds, and both 1,4 and 1,6 glycosidic bonds are only present in amylopectin and glycogen. Hence the answer is B, 1 and 4. Question 8. The diagram shows a small part of a polypeptide. What would the products be if the part shown was completely hydrolyzed? Now hydrolysis is the addition of water to break the peptide bonds, whereas condensation is the reverse. It is the process of removing water to form peptide bonds. Since they are asking for the part being completely hydrolyzed, water will be added, and when water is added, OH will be taken by C double bond O and H will be taken by NH. And as you can see, I have labeled over here where water will be added, and when water is added over there to these two regions, 3 products will be formed, and the most closest and correct representation of those products is option D. Question 9. The enzyme trypsin hydrolyzes proteins to amino acids. Trypsin does not function when the pH is very low, as its 3D shape would be changed. What explains this change in 3D shape? A. Hydrogen ions attach themselves to negatively charged R groups. B. Hydrogen ions disrupt disulfide bonds. C. Hydrogen ions increase hydrogen bonding between amino acids. D. Hydrogen ions reduce the affinity of hydrophilic R groups for water. Now option D is obviously incorrect, as hydrogen ions has nothing to do with the affinity of hydrophilic R groups. Option C. Hydrogen ions increase hydrogen bonding. That's also incorrect, as hydrogen ions do not participate in hydrogen bonding, but rather hydrogen bonding occurs between strongly polar groups such as NH, CO and OH groups. B. Hydrogen ions disrupt disulfide bonds. That is also incorrect, as disulfide bonds are the strongest bonds and you need to boil in order to disrupt these bonds. Option A. Hydrogen ions attach themselves to negatively charged R groups. Now, as you know, hydrogen ions are positively charged, and R groups can be of various types. They can be neutral, they can be positively charged, and they can be negatively charged R groups. Therefore, since they mentioned about the pH being very low, pH decreases, then hydrogen ions increases. Since hydrogen ions are positively charged, it binds with negatively charged R groups, as opposite charges attract. Therefore, the answer is A. Question 10. The flowchart shows some of the steps in the formation of collagen. Which row correctly identifies X, Y and Z? They have started the flowchart from 3 polypeptide chains held together by X bonds to form triple helix. Now in collagen, 3 chains are held together by hydrogen bonds. Therefore, the options are between option B and option C. If we go further down the flowchart, triple helix held together by intermolecular bonds to form Y. Now triple helix along with other triple helices form fibrils. Hence Y should be fibrils. And when fibrils lie alongside other fibrils, they form fibers. Therefore, the answer should contain hydrogen bonds leading to triple helix, which leads to fibrils, and therefore lastly fibers. Hence X should be hydrogen, Y should be fibrils, Z should be fibers. Therefore the answer is B. Question 11. Which molecules contain at least 2 double bonds? 1. Saturated fatty acid, 2. Collagen, 3. Hemoglobin. Now saturated fatty acid doesn't have any double bonds. Only unsaturated fatty acids will have double bonds. Therefore option 1 is obviously incorrect. Hence if we look at the answers, option A, B and C has 1. Only option D doesn't have 1. Therefore the answer must be option D. Collagen and hemoglobin. Question 12. 2 experiments X and Y were carried out using an enzyme from humans. Experiment X was carried out at a constant temperature of 37 degrees Celsius. During experiment Y, the temperature was increased from 37 degrees Celsius to 80 degrees Celsius. All other factors were kept the same. Which graph shows the results? 2 experiments were carried out using the same enzyme from humans. However, the experiments were kept at 2 different conditions. One was kept at 37 degrees Celsius, which is a normal body temperature, and the other is kept at 80 degrees Celsius, which is a very high temperature, hence leads to enzyme denaturation, as enzymes do not work at such high temperatures. They are asking which graph shows these results. And if you look at the graphs, the x-axis contains time in minutes and the y-axis contains product concentration. One thing you should know is that the product concentration doesn't drop to 0 even if the reaction stops, as some made is left behind. Therefore, option B is obviously incorrect as it drops to 0. Option D is also incorrect as product concentration drops in Y. Option C is incorrect as Y occurs at a higher temperature, hence it cannot have a faster rate of reaction. It should be less than the rate of reaction of X. Hence, the best suitable answer is option A. Question 13. What affects the rate of an enzyme catalyzed reaction when in the presence of a non-competitive inhibitor? They are asking what affects the rate of an enzyme catalyzed reaction, and they have mentioned non-competitive inhibitor. They have given the options as 1. Enzyme concentration, 2. Inhibitor concentration, 3. Substrate concentration. Factors affecting enzyme catalyzed reactions are enzyme concentration, substrate concentration, inhibitors, then there is temperature and pH. Therefore, if you go through these options, option 1, 2 and 3, all of them do affect the rate of an enzyme catalyzed reaction, even if it's non-competitive or competitive inhibitor. Now you may be wondering, how does substrate concentration affect since it's a non-competitive inhibitor? Well, some could be inhibited and some cannot be inhibited, as not all enzymes will be inhibited by the non-competitive inhibitor. Therefore, it does affect the rate of an enzyme catalyzed reaction. Hence, the answer is A, 1, 2 and 3. Question 14. What describes the fluid mosaic model of membrane structure? If you recall the definition of fluid mosaic, fluid means the proteins and phospholipids can move within their own monolayers, and mosaic means that the proteins are arranged in a scattered pattern. Therefore, if you go through the options, A, cholesterol makes a membrane more fluid. That doesn't describe the fluid mosaic model. B, fluids can cross the membrane by diffusion or osmosis. That also doesn't describe the model. C, proteins act as receptors. That is also incorrect. D, proteins and phospholipids move and change places. Yes, that represents the fluid nature of the model. Hence, the answer is D. Question 15. Blocks of agar are stained with a pH indicator and used to investigate the diffusion of an acid solution. Which block would completely change color the fastest? In order for the block to change color the fastest, it should have the highest surface area to volume ratio. As higher the surface area to volume ratio, faster is the diffusion. If you go through these options and calculate the surface area to volume ratio, option B will give you the highest surface area to volume ratio. Hence, higher the surface area to volume ratio, faster the diffusion, faster the color change. Therefore, the answer is B. Question 16. The diagram shows the water potential of three adjacent plant cells P, Q and R, which shows the net movement of water between cells P, Q and R. P represents minus 198 kilopascals, Q represents minus 224 kilopascals, R represents minus 212 kilopascals. Now, out of all these three, which one has the highest water potential? In order to find that, you should check which one has the lowest negative number. Lower the negative number, higher the water potential. Hence, P has the highest water potential. Now, if you recall the movement of water, that is osmosis, osmosis occurs from a region of higher water potential to a region of lower water potential. That is down a water potential gradient. Hence, water will move from where there is higher water potential to lower. Since P has the highest water potential, it should start from P, followed by R, which has lower water potential than P but higher water potential than Q, and the least water potential present is in Q. Hence, the water movement will be from P to Q as well as P to R and R to Q. Therefore, the answer is A. Remember, more negative the number, lower is the water potential. Question 17. Which row correctly describes parts of chromosome structure present during mitosis? They have given the columns as centromere, chromatid, telomere. Now, centromere is a region where there are no genes and has a region of non-coding DNA, as well as it holds two chromatids together. Chromatid contains a DNA sequence coiled around histone proteins. Telomere has a protective function that protects the ends of a chromatid during DNA replication. If we go to the options A, B, C and D, option A is incorrect as it mentions centromere as a region of a chromosome with no DNA. However, centromere is of a specialized DNA sequence that is called non-coding DNA. Therefore, option A is incorrect. D. Region of non-coding DNA, correct. Double-stranded DNA molecule with histone proteins, correct. Region of DNA with many short repeated sequence of bases, yes, that is also correct. Hence, option B is the correct answer. If we go to option C, they have described telomere as region of DNA with no proteins. However, telomere does have proteins associated with production of telomerase, which is an enzyme. Therefore, option C is incorrect. If we go to option D, they have mentioned that under centromere, region that attaches to spindle microtubules and divides during prophase. Centromere division doesn't occur during prophase but rather during anaphase. Hence option D is also incorrect. Therefore, the answer is B. Question 18. The diagram shows the relative time taken for each stage in the cell cycle. Which row identifies the correct sequence of the stages in the cell cycle? This diagram right here represents the relative time taken. And a key thing you should know is that interphase is the most time taking process, followed by mitosis, and cytokinesis takes the least time. Why does mitosis take more time than cytokinesis? Well, mitosis has many phases. It has prophase, metaphase, anaphase as well as telophase. Therefore, it takes more time than cytokinesis, where cytokinesis is just cell division to produce two daughter cells. Hence, X should be mitosis, Y should be cytokinesis, Z should be interphase, which is represented by option C. Question 19. Scientists have made a nucleic acid, HNA, that has a sugar with the same number of carbon atoms as glucose instead of deoxyribose. Although genetic information can be stored by HNA, naturally occurring DNA polymerase cannot replicate HNA. Which statements could explain why naturally occurring DNA polymerase cannot replicate HNA? 1. DNA polymerase cannot form bonds between the sugars of two HNA nucleotides. This is a wrong statement as DNA polymerase has nothing to do with sugars. Option 2. DNA polymerase cannot form hydrogen bonds between two HNA nucleotides. That is an incorrect statement as DNA polymerase has nothing to do with hydrogen bonds. Option 3. HNA nucleotides do not fit into the active site of DNA polymerase. Yes, that could explain why naturally occurring DNA polymerase cannot replicate HNA, as even though it's of the same number of carbon atoms as glucose, it could be of a slightly altered shape. Option 4. The shape of an HNA nucleotide is slightly larger than that of a DNA nucleotide. Yes, that could also explain why the DNA polymerase cannot replicate HNA. As I have mentioned, it could be of a different shape. Therefore, the answer is D. Option 3 and 4 only. Question 20. Which statements about the nucleotide containing uracil are correct? 1. Uracil is a pyrimidine. 2. The carbohydrate is always ribose. 3. Base pairing occurs with 3 hydrogen bonds. Option 3 is obviously incorrect as base pairing of uracil is with adenine and A bonds with U using double bonds, that is only 2 hydrogen bonds and not 3. Since option A, C and D contain 3 as the answer, that leaves us with option B being the right answer as B doesn't have option 3 in it. 1 and 2 is correct because yes, uracil is a pyrimidine as only guanine and adenine are purines, the rest are all pyrimidines. 2. The carbohydrate is always ribose. Yes, that is also correct as uracil is associated with RNA and not DNA. And the main component of RNA is nothing but ribose. Hence, the answer is 1 and 2 only, that is option B. Question 21. DNA replication involves several stages. 1. Each strand of DNA double helix acts as a template for the opposite strand. 2. The enzyme DNA polymerase links bases together. 3. Hydrogen bonds between bases A and T and between C and G are broken. Which statements about DNA replication are correct? If you look at option 2, the enzyme DNA polymerase links bases together, that is incorrect as DNA polymerase links nucleotides together and not bases, and that rules out option A and B. Considering between option C and D. Option C and D both have 3 in it. Hence, let's look at 1. Each strand of DNA double helix acts as a template for the opposite strand. Yes, that is correct. During replication, double strand unwinds or unzips, leading to each strand acting as a template for a new strand to be formed. Therefore, option 1 is also correct. Hence, the answer is C, 1 and 3 only. Question 22. The sequence of bases in mRNA for the first 8 amino acids in the beta polypeptide of adult hemoglobin is shown. In hemoglobin C, which is a cause of hemolytic anemia, the sequence is given. The coding for 7 of the amino acids is listed in the given table. Which change occurs to the amino acid sequence of adult hemoglobin to make hemoglobin C? First of all, the sequence of bases is given in the form of mRNA. Therefore, we have to convert it to the DNA triplet. Therefore, if we convert the given adult hemoglobin sequence to DNA triplet, we get the sequence shown in red, which I have mentioned above. Now, in this given DNA triplet, if we compare it with the hemoglobin C DNA triplet, only the underlined three bases change. Therefore, the change is represented by CTC changing to TTC. Therefore, glutamic acid is changed to lysine. Hence, the answer is C. Question 23. Some antibiotics kill prokaryotes by binding to RNA polymerase. What effect will this have on protein synthesis? RNA polymerase join RNA nucleotides together to form phosphodiester bonds during condensation reactions. Therefore, if we go through the options A, B, C and D, option B represents the correct answer. Condensation reactions joining RNA nucleotides will not take place, as the main function of RNA polymerase is to join adjacent RNA nucleotides together during condensation reactions. Hence, the answer is B. Question 24. The diagram shows transverse section through parts of plants. Which of the labeled regions contain cells which are dead? Now, cells which are dead are only present in xylem vessels. Hence, the answer is answer A, as answer A represents xylem. All the others represent phloem. Question 25. The diagram shows a distribution of tissues in part of a transverse section through a plant organ. Now, I have labeled the parts of the transverse section, the outer layer being the epidermis, followed by the cortex, followed by the endodermis, and the star-shaped patch represents the xylem, and the small circles represent the phloem. They are asking which row correctly identifies tissues 1, 2 and 3. 1 is cortex, 2 is endodermis, 3 is phloem. Hence, the answer is A. Question 26. The circumference of a tree stem was measured continually for 48 hours. The results are shown on the chart recording. What explains the changes in circumference recorded during the 48 hours? Circumference change of a tree is related to the tension forces, and tension forces changes throughout the day. Hence, if we go to the options A, B, C and D, option B talks about the tension forces which increase during the day. Hence, the answer is B. Question 27. Which description of movement in phloem is correct? Option A. Adding sucrose to a sieve tube element increases its water potential so that water enters and increases the hydrostatic pressure. That is incorrect, as when a sucrose is loaded to a sieve tube element, the water potential decreases, leading to water entering. Therefore, it doesn't increase but rather decrease. Hence, option A is incorrect. If you look at answer B, at a sink such as a storage organ, sucrose is removed from a sieve tube element and polymerized into starch. Sucrose cannot be polymerized to starch, as starch is a polymer of alpha glucose, whereas sucrose is made up of glucose and fructose. Therefore, option B is incorrect. Option C. At a source such as a photosynthesizing leaf, sucrose enters a sieve tube element by facilitated diffusion. That is also incorrect, as sucrose enters by active loading by the plant, as it requires ATP and not facilitated diffusion. Hence, option C is also incorrect. That leaves us with option D. At a source, sucrose is loaded into a companion cell using a protein that carries both hydrogen ions and sucrose molecules. Yes, and that protein is called the cotransporter molecule. Hence, the answer is D. Question 28. Which statements about blood vessels are correct? 1. Collagen fibers in the walls of arteries and veins prevent overstretching. 2. Elastic fibers in large arteries allow stretching during ventricular systole. 3. Smooth muscle in arteries controls the distribution of blood to different parts of the body. 4. Veins have valves because their walls are thin. Option 4 is obviously incorrect, as veins do not have valves because the walls are thin, but they have valves because the pressure inside veins is very low. Hence, in order to prevent the backflow of blood, veins have valves. Therefore, if we go through the options A, C and D, all three have option 4 in it. Therefore, they are all wrong. The correct answer should be option B, 1, 2 and 3 only. Question 29. The photomicrograph shows 3 white blood cells labeled X, Y and Z. Which row correctly identifies these cells? Now, I have labeled over here how you describe the nucleus as it appears. So, Y is of a circular nucleus, X is of a lobed nucleus, Z is of a kidney-shaped nucleus. Now remember, X, since it is a lobed nucleus, it represents a neutrophil. Circular nucleus means that it is a lymphocyte. Kidney-shaped nucleus represents a monocyte. Therefore, the answer is D. Cell X being a neutrophil, cell Y being a lymphocyte, cell Z being a monocyte. Question 30. The enzyme carbonic anhydrase catalyzes the reaction carbon dioxide plus water gives hydrogen carbonate plus hydrogen ions. Which statement describes the role of the enzyme carbonic anhydrase? 1. To speed up the decrease in pH of blood in the presence of carbon dioxide. 2. To facilitate the Bow effect in hemoglobin. 3. To speed up the reaction between carbon dioxide and water. If we look at option 1, to speed up the decrease in pH of blood in the presence of carbon dioxide, yes, carbonic anhydrase acts when there is a high partial pressure of carbon dioxide and it catalyzes the reaction between carbon dioxide and water. Hence, option 1 is correct, as when it catalyzes the reaction between carbon dioxide and water, hydrogen ions are released. Therefore, hydrogen ion concentration increases, leading to pH decreasing. If you look at option 2, to facilitate the Bow effect in hemoglobin, what do you mean by Bow effect? Bow effect is due to the presence of high partial pressures of carbon dioxide causing hemoglobin to release oxygen. Again, high presence of carbon dioxide leads to the enzyme catalyzing the reaction between it and water. Hence, as the enzyme is involved, option 2 does describe the role of the enzyme. If you look at option 3, to speed up the reaction between carbon dioxide and water, carbonic anhydrase is an enzyme and all enzymes do speed up the rate of a reaction. Hence, the answer should contain all 1, 2 and 3. Therefore, the answer is A. Question 31. The graph shows the oxygen dissociation curves of adult hemoglobin at two partial pressures of carbon dioxide, W and X. Which pair of statements are correct? They have plotted the x-axis as partial pressure of oxygen in kilopascals and the y-axis as the percentage saturation of hemoglobin with oxygen, and they have drawn the two curves. Obviously, curve W lies towards the right side compared to curve X. Hence, curve W has a higher partial pressure of carbon dioxide. Now, if we look at the statements given, statement 1, curve W shows the oxygen dissociation curve at a higher concentration of carbon dioxide than curve X. Yes, as I mentioned, curve W lies to the right hand side of curve X, hence leading to the fact that it has a higher partial pressure of carbon dioxide. As when a curve shifts to the right hand side, that implies that there is more carbon dioxide present. Therefore, option 1 is correct. If you look at statement 2, curve X shows the oxygen dissociation curve at a higher concentration of carbon dioxide than curve W. That is incorrect, as only curve W shows at a higher concentration of carbon dioxide. Statement 3, at a partial pressure of oxygen of 7 kilopascals, the hemoglobin from curve W has a higher affinity for oxygen than hemoglobin from curve X. That is incorrect, as if we look at the graph, curve W at 7 kilopascals has a lower percentage saturation of hemoglobin, hence it cannot have a high affinity towards oxygen. Therefore, that leaves us with the options being 1 and 4. If we go through statement 4, at a partial pressure of oxygen of 7 kilopascals, the hemoglobin from curve X has a high affinity compared to curve W. That is correct, as curve X at 7 kilopascals, it has a higher percentage saturation if we look at the y-axis. Hence, the answer is B, 1 and 4. Question 32. A person has two blood tests 1 month apart. The number of each type of cell in a fixed sample size is counted. What could this suggest about the person based on the results after 1 month? The columns given are body temperature higher, move to higher altitude, ATP synthesis in cells is higher. Now, after 1 month, both red blood cells and lymphocytes are higher. Red blood cells being higher implies that the person has moved to higher altitude, as the RBC count increases when a person moves to a higher altitude due to reduced partial pressures of oxygen. In order to compensate for the reduced oxygen partial pressure, RBC count will increase. Therefore, we can say that the person has moved to higher altitude. That leaves us with options B and D. Lymphocytes increases after 1 month. Hence, antibody levels and cytokine levels increases, as lymphocytes are of two types, B lymphocytes and T lymphocytes. Now, energy is required to make these proteins, as antibody and cytokines are both proteins. Hence, ATP synthesis should be higher. Therefore, that leaves us with option D being the right answer. Question 33. A student was asked to describe the differences between 4 microscope slides of sections taken from different parts of the gas exchange system. Which is the correct identification of the parts of the gas exchange system? A key thing we should remember over here is that cartilage is only present in two structures, that is in the trachea in the form of C-shaped rings and in the bronchus in the form of irregular blocks or plates. Now, A is incorrect, as slide 2 mentions cartilage being present, whereas the answer given is bronchiole, which doesn't have any cartilage. Therefore, we can rule out A. Answer D is also incorrect, as slide 1 says cartilage is not present. However, the answer given is bronchus, which does have cartilage. Hence, we should consider between options B and C. Considering between B and C, C is the correct choice, as slide 2 talks about cartilage being present in the form of rings, which is only present in the trachea and not in the bronchus. Therefore, the answer is C. Question 34. Cigarette smoke contains carbon monoxide, nicotine and tar. Which row correctly describes how these substances affect the body? If we look at answer A, carbon monoxide stimulates nerve endings. That is incorrect, as carbon monoxide doesn't have anything to do with nerve endings. If we look at option B, carbon monoxide forms carbaminohemoglobin. That is also incorrect, as carbon monoxide forms carboxyhemoglobin. If we look at answer C, carbon monoxide causes increased blood pressure. That is incorrect, as only nicotine leads to increased blood pressure. That leaves us with D being the only choice. Hence, D is the right answer. Carbon monoxide combines with hemoglobin. Nicotine stimulates nerve endings. That is correct. Tar is carcinogenic, yes, as it has carcinogens which can lead to obstructive lung disease. Hence, the answer is D. Question 35. Chronic obstructive pulmonary disease includes chronic bronchitis and emphysema. In what order do these events occur as emphysema develops? I have labeled towards the side which one occurs first and which one goes last. So first it starts with tar paralyzing cilia. Second, bacteria accumulate in the alveoli. Third, macrophages secrete elastase. Fourth, macrophages move into alveolar air spaces. Therefore, if we follow that order, the only answer we get is answer B. It is from 2 to 4, 4 to 3 and 3 to 1. Question 36. If someone smokes cigarettes, what will be the immediate result of this action on the red blood cells? Over here they are asking for the immediate result, and the only immediate result that occurs is that less oxyhemoglobin will form. Hence, the answer is C. Question 37. What is correct for tuberculosis? Tuberculosis is caused by Mycobacterium tuberculosis or Mycobacterium bovis, which are both a bacteria. Tuberculosis is also an aerosol or droplet infection, hence is airborne transmission, and it doesn't include any sort of a rash. Hence, the answer is A. Question 38. Some common antibiotics are listed. The action of each antibiotic is described. Which of these antibiotics will affect the activities of bacterial cells only? Now, bacterial cells are only affected by option 4, as peptidoglycan is present in the bacterial cell wall and not in any other cell walls. Therefore, the answer is 4 only, which is D. Question 39. Which method of gaining immunity can be described as natural active immunity? A. Feeding on colostrum. B. Inhaling the chickenpox virus. C. Injection with antibodies. D. Through the placenta. Options A and D are both natural, however passive immunity. Comparing between B and C, C. Injection with antibodies is artificial and passive. Therefore, that leaves us with B. Inhaling the chickenpox virus, described as natural active immunity. Question 40. Which statements correctly explain why smallpox has been eradicated but not malaria or cholera? 1. Cholera vaccines provide only short-term immunity. 2. Plasmodium antigens change during the life cycle. 3. Smallpox antigens remain stable. 4. Vaccines only work against viruses. Now obviously, statement 4 is incorrect, as vaccines work on both bacteria as well as viruses. If we consider between answers A, B, C and D, answers B, C and D, all three of them contain statement 4. Therefore, that leaves us with A being the right answer. 1, 2 and 3 only. And there we have just completed the 40 questions. Thank you for watching till the end. If you would like more content, do keep watching more of my videos. If you would like me to do any specific paper, let me know in the comment section below. Until then, stay tuned for more.